Okay, let's start. Okay, good morning, afternoon, evening. Hola, hello, bonjour. Hola, ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 57th seminar, and I call it What's You Day. I am traveling mostly in Asia right now, and I am now in Singapore after giving 4 talks since last Saturday. I will continue to take trips 2 more weeks to give a keynote and a couple of more talks. Here is a little bit late, as you know, 11 p.m. And however, I am so excited to have 2 speakers today. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Himadri Pakrasi. He is a professor of the biology department at WashU and my faculty mentor here, especially helping me tremendously when I was a junior faculty. He cares about young faculty a lot, helping many of my colleagues as well. He's also a pioneer in the cyanobacteria research field, as many of us know. So today, I also heard our rising star speaker just, join as a assistant professor, and Himadri always kind of care about those kind of junior faculty. So this is great match, I would say. And then Himadri, thank you so much for your time today. And then the virtual podium is all yours now. Thank you.
Thank you, Taisek and Christian. So nice to meet you. Taisek, first of all, congratulations in keeping this thing going. 57, my God. You have spent a significant part of your life actually in taking care of this seminar series. And I keep on hearing from various parts of the academic community as well as in some in industry, how enjoyable this series has been. What I would like to do now is just to introduce myself, the kind of things that interest me in the area of synthetic biology and systems biology, by sharing with you a few slides. So let me put the slides up. And can you see the slides okay? Okay, so I have been at Washington University in St. Louis for more than 3 decades. And in all this time, the principal process that we study is oxygenic photosynthesis. And this is kind of a cartoon diagram just to remind ourselves about the green process that nature has devised. And we want to actually take advantage of this natural green process to create photosynthetic cell factories, okay? So cyanobacteria, they are non-model microbes, and then they hold enormous potential for use as green E. coli. And one aspect that has interested us over the last decade or so is to try to figure out if we can get some cyanobacteria that can grow fast. I don't have to say any more as to why fast-growing organisms are important for bioproduction. When we started this, there were some standard model strains of cyanobacteria and algae that were available, and they grew, reasonably fast, but not so fast. At least not as fast as our lab members really wanted them to be. So at that time, we said, okay, use of fast-growing cyanobacterial cell factory are highly desirable. And during that process of looking around and trying to find organisms that already exist in nature, and then going through lab-based evolution process, we came out with this strain that's now called Synechococcus 2973. It's in the Utex culture. And without going through all of this data, basically, this is the important thing. Synechococcus can grow as well as Saccharomyces cerevisiae with sugar, whereas Synechococcus does not need any of that. It's just CO2 and light, 1.5-hour doubling. Not as fast as what E. coli is purported to be, but it has actually changed the paradigm, I would just say, because this strain is now being used in many places. Christian, I know that you are interested in collaboration. In our entire process of work, I have been collaborating over 12, 13 years now with Professor Costas Maranas at Penn State. And here is, I refer to a review paper that came out through our collaboration that actually addresses what probably will be something that you will be talking about, is metabolic model guided strain design. And only one piece of data that I will show you is that by using that approach and using this fast growing strain, we were able to have outstanding and rates of sugar production by this cyanobacterium. This is a hot area. A lot of people are working on this. The idea is that the cyanobacteria will be making the feedstock for other specialized microbes that will be making the desired products. In this kind of co-culture process, this kind of sugar production ability is very, very important. And as you can see, we can get 2 grams per liter per day, which may translate into numbers that I don't show here, is comparable or better than what sugarcane does. Although there are other challenges, I must acknowledge that, because large-scale production of algae and cyanobacteria is a big challenge. So that's all I wanted to show you at this stage, just to introduce what we do. And I'm really looking forward to hearing in person now what, Christian, you are going to say. I have to go teach in about 15 minutes or so. And so I will then come back and listen to the recorded version. That's all that I wanted to say.
So fantastic, Himadri. It is amazing talk. It's fantastic. So, I mean, to solve lots of problems, especially global problem, including climate crisis. Obviously, cyanobacteria is one of the most important organism, and then your pioneering work is always, respected and also appreciated. And I learn a lot from you. I mean, so I want you to kind of continue to pioneer those fields, and then potentially making cyanobacteria even faster than E. coli one day. So that's my hope.
Okay, thank you. Yeah, hopefully that will happen. If we don't do it, somebody like Christian is going to do it, because we just hand over our knowledge to the next generation, right?
Yeah, because we are rely on those young people, because they are our future leaders. So obviously, Christian, you have lots of things to do in the future. We giving a lot of pressure to you. All right. Okay. So thank you so much, Himadri. That's brilliant talk. I love it. Okay. Now the main speaker of today, with a longer introduction, Dr. Christian Euler received his PhD at the University of Toronto while leading activities at the company he co-founded, SynBYSS Biotechnologies. His career goal is to find an academic position in which he could continue his involvement with SynBYSS. And in fact, as I mentioned earlier, he secured assistant professorship very recently, and then he will start his position next year. So congratulations, Christian.
Thank you.
Yep. Christian's research interests are centered on metabolic regulation, including the thermodynamics of regulatory systems and analysis and design of enzyme-level regulatory structures. His work has uncovered the fundamental design principles and constraints of system-wide enzyme-level regulation in metabolism. He is excited about using existing synthetic biology tools and developing new synthetic biology tools to answer fundamental questions about the origin of life, evolution, and for design and optimization of metabolism. So, Christian, thank you so much for joining today. And then also, thank you for, sharing your, idea, vision, also your data, next, 30 to 40 minutes, and please take it away. Thank you so much.
Great. Thank you. Thank you. So I'm just going to share my screen now. You can see?
Yep.
Great. Okay. So yeah, thanks to you guys for your time, for everyone who's here, and for having me. Today I'm going to present really what kind of makes up my PhD thesis, which I just recently successfully defended, as mentioned, at University of Toronto. So I've titled this presentation, Metabolites as Flux Signals, Underlying Mechanism and Regulatory Structure. This might be a bit different from the other talks in this series in that the meat of this is really going to be around really more of a systems biology topic and really about fundamentals. So not necessarily building new circuits or understanding new circuit design, but uncovering some fundamental aspects of sort of regulation. But I will bring it back to, what does it mean in terms of translating that in terms of synthetic biology at the end of the presentation. So here's just kind of an overview. I'll talk about the background and the research question. There's three kind of pieces to this story that I'll bring out. One of them involves sort of the idea of flux sensing, how that relates to thermodynamics. Then we have sort of the regulatory network that is formed by small molecule regulatory interactions. And then I'm going to take like a sort of an informational perspective to this kind of regulation to understand the constraints and the optimization of small molecule regulation. And then finally, like I promised, I'll bring it back to synthetic biology and understand how my work can help us develop new tools to optimize engineered metabolisms. So I'm not going to introduce myself too in depth because I've already had that introduction, but I just did want to mention or highlight that I've sort of worked. I'm going to be talking, my research is sort of more systems biology oriented, but I am going to be, as part of my company, Ficus Biotech, I've been working for the last 2 years to scale a SynBYSS platform to 1,000 liters, and we're going even beyond that for production of a chemical using an E. coli chassis. So I do have that sort of SynBYSS background as well to inform what I'm doing. Great. So let's dive in. And first of all, to understand the scope of the work I'm presenting today, it's really important first to understand the idea that metabolic regulation is hierarchical. So, we often think in terms of gene circuits, gene networks, and how those turn expression on or off, basically. And we often represent that sort of, in this way with sort of a gene promoter, this kind of structure. But the reality is once enzymes or other proteins really, but we'll focus sort of on metabolic enzymes today, once they're produced, their activity is also directly regulated by molecules such as metabolites, signaling molecules. And in recent literature, this kind of regulation has been called small molecule regulation, or SMR. It includes what we think of as allosteric regulation, but also other forms of inhibition, direct competitive inhibition, uncompetitive, and that kind of thing. this has kind of been lumped together. All these mechanisms have sort of been lumped together because at the end of the day, what this kind of regulation does is change fluxes. So if an enzyme is catalyzing this reaction, A to B, let's say the product of that reaction might inhibit that flux, regardless of how the mechanism sort of works. Now, small molecule regulation, or SynBYSS, is not really well understood from a broad sort of systems level perspective. It's been characterized from a biochemical kind of reductionist viewpoint for as long as we've kind of understood it, and that is, we take the enzyme, we put it in a tube, we add the one inhibitor, and we see how it changes the enzyme activity. But there's been very little work until my thesis, or there had been very little work until my thesis, about how do we organize those interactions sort of at a metabolic level, and what's the global picture of how these interactions change fluxes or control fluxes, and why are they organized the way they are. It's really kind of important because, if we want to bring these metabolic pathways to scale, and this is a 1,000-liter reactor that I work with on a regular basis, we really need to understand how metabolism is stabilized, how fluxes are controlled, so that we can do sort of precision balancing of those fluxes. And in particular, if metabolic engineering is going to tackle this grand challenge of carbon fixation, we need to really understand how autocatalytic, or sometimes called anabolic cycles, are regulated and stabilized. And the small molecule regulation, as you'll see, probably plays a role in that. And so understanding these things is really important to be able to bring systems to scale. Even though, my talk is going to be quite fundamental, we should keep this in mind as we go through. So for me, the high level sort of relevant research question was, why does small molecule regulation exist in the metabolic network? Of course, I mean, this is very broad. It's too broad to be answered in a single dissertation. But with the available information and the available approaches, and maybe some novel approaches as well that we'll talk about, we can kind of break this into two sub-questions. So first of all, which metabolites make good signals in small molecule regulation? So we might see patterns of certain metabolites being used to regulate enzymes, and the question might be, why is that? What about their sort of positioning in the metabolic network makes them good signals? And then how does small molecule regulation function to control fluxes? So how do we organize those regulatory interactions to make sort of decisions, one could say, about how flux moves through or changes in the network given some external conditions? So in answering the first question, I actually looked to fructose 1,6-bisphosphate, or FBP, as a model, because we know that FBP is sort of an almost global small molecule regulator of enzyme activity. There are a lot of examples of FBP activating downstream enzymes in glycolysis. So for example, in E. coli and yeast, pyruvate kinase 1 is activated, it's activated by FBP. But many downstream enzymes actually are activated by FBP. And in looking at this as a model, I learned that FBP is a known flux sensor. This means that as the glycolytic flux increases, the steady state concentration of FBP also increases. So the concentration of FBP is a good signal for the glycolytic flux. And so this kind of, led me to the question, can we ask, are flux sensors good regulators in general? And so now to answer this question, I really had to kind of get into how flux sensing itself arises. And as we'll see, this kind of gives us a clue as to which metabolites make good sensors or good regulators in the network. And so, to sort of visualize or understand the constraints that lead to good flux sensing, we can kind of think of just a very simple conversion of a substrate to a product by an enzyme with some, rate of arrival or rate of generation U. And when this reaction is thermodynamically favorable. So it likes to go in the forward direction. And that's given by, a K-eq much greater than 1, okay? Or you can also think about a standard Gibbs free energy much less than 1, much less than 0. So when this is the case, the rate at which the substrate is consumed is a function of that concentration. Because we know Michaelis-Menten kinetics, even mass action kinetics, as S accumulates, the rate at which it is consumed will be a function of that. But in the case in which this reaction is favorable in the reverse direction, where we have the equilibrium constant less than 1 or the Gibbs free energy greater than 0, we actually have to accumulate a lot of substrate in order to drive against this sort of thermodynamic barrier to move this reaction in the forward direction. And in this case, of course, with saturation kinetics, as this substrate has to build up to drive the flux forward, we can approach the maximum rate at which this enzyme can work. And so in this case now, because this rate is constant, the steady state concentration of S will become sort of a function of this upstream flux U. And so this gives us an idea of where flux sensing might arise. When this reaction is thermodynamically unfavorable, we can expect the substrate to accumulate as a function of the upstream flux. And so often I will use these symbols. So I'm just going to introduce them. So the steady state concentration, I've noted that with an asterisk, S star, and U is the incoming flux, V is the downstream flux here. So for all the engineers in the crowd, this is very similar to how a Venturi meter works. I like to think in sort of, physical analogies. And so in a Venturi meter, we have flow coming into a pipe and it's choked. And as a result of that choke point, the material accumulates upstream, which creates a pressure differential. And with the pressure differential, we get this different, the readout is a difference in heights of fluids. And with that difference in heights, we can measure the flow going through this pipe. And the analogy is very similar. So in the Venturi meter, the flow is given by Q. It's the rate at which fluid is entering. In our system, this is U, the upstream flux. In the Venturi meter, we get a readout, which is a difference in pressure or pressure differential. In the metabolic system, it's how far we are from equilibrium, given by this sort of K-eq value. And then the degree to which the flow is choked is given by, again, this equilibrium value, whereas it's given by the area of the ratio of the cross-sectional areas in a Venturi meter. And so we use Venturi meters to measure flows. And this sort of system, this sort of setup is very analogous to a Venturi meter where you might be able to get a flow readout. It's really important because, of course, flows have to be calculated. Flows we get from a certain a certain event happening over time. And so we need to do a little division to get a rate. If we can directly measure some signal that gives us a rate, that's really useful. And that's what a Venturi meter does. It's what the speedometer in your car does. And it's maybe what the concentrations of these sort of flux sensing metabolites can do for us. Oops. So we can kind of understand this by just, again, looking at our very simple enzyme catalysis system and just adjusting the thermodynamic parameter and calculating some steady state concentrations to understand how the steady state concentration is related to the upstream flux for various sort of parameter situations. So in the irreversible case, you can see we have to operate, very close to the maximum, and this is a scaled flux. So 1 here is the maximum. We're approaching Vmax of that downstream enzyme, which is why we get really quite extreme accumulation. But now, as we decrease the value for the equilibrium constant, we make the reverse reaction more favorable. You can see that we get much stronger accumulation for a lower upstream flux, and it's also much more linear. So we kind of have this parameter I've just described as the amplification, the signal amplification. The minimum value is the slope, related to the slope at the zero flux. So for really what I'm going to describe as thermodynamically constrained reactions, so those which are really favorable in the reverse direction compared to the way they function in metabolism, this amplification is proportional to the negative of the logarithm of that k value. And so as we decrease, we get a much more linear signal, which is great. In control, we would like a linear signal. As this flux changes very slightly, we get a large change in the steady state substrate concentration. And that gives us a signal that we could use to measure and read out that flux. So this really means that there's a threshold sort of thermodynamic constraint value where that could give us flux sensitive substrate concentrations. So to find this threshold for E. coli, I sampled all known enzyme kinetic parameters, and again, modeled it just with this very simple substrate to product with some incoming flux. And I found that, a randomly selected reaction with a standard Gibbs free energy above about -4 kilojoules per mole is 50% more likely, is more than 50% likely to have flux sensitive substrates. So, yeah. And so you can see that's kind of reflected here as the standard Gibbs free energy, which, we don't have to use that. We can also use the equilibrium constant. But if we sample the kinetic parameters for sort of all the enzymes that we know in E. coli and generate these minimum amplitude or amplification curves, you can see that sort of around this value of -4, we start to get a really strong amplification. And that's sort of 50% in blue here. And then for 90% of possible parameter combinations, we have sort of this value up here. So that gives us bounds, like a median estimate and an upper bound estimate for where flux sensitivity sort of will occur in the network. And so for median values, again, this Gibbs free energy is around, which I'm going to call the maximum downstream Gibbs free energy, is around -4 kilojoules per mole. And so to sort of then actually test this value, I took fluxomic and metabolomic data for E. coli across 14 different native carbon sources from 3 high quality data sets and correlated all measured concentrations against all fluxes. And then found those metabolites which have a strong, whose concentrations have a strong correlation with some upstream flux. We can characterize that with a Pearson R value, of course, very simple. I'm sure we're all familiar with how that works. And what you can really see is that, again, as reactions become more favorable in the reverse direction, the probability that they are correlated, that the metabolite concentrations are correlated with some upstream flux becomes very high. And so the threshold value of around -4 is where we see sort of this jump. This is just the sort of rolling average of these Pearson R values. And some very familiar metabolites also appear. So for example, this process showed that, in fact, yeah, FBP is correlated to glycolytic flux, which is great. And it fits with sort of what we understand the most reverse favored consuming reaction of FEP has sort of a Gibbs free energy around 20 kilojoules per mole. But there's also a bunch of other metabolites that appeared that could serve us as flux sensors and that have not really been characterized too well as such in the data, including malate, 6-phosphogluconate, citrate, and isocitrate, and then some pentose phosphate pathway intermediates, ribulose 5-phosphate, ceduloheptose 7-phosphate, and connotate here. So this is really exciting because it kind of showed what I was able to show with the modeling, that there is a threshold above which there seems to be strong correlations between steady state concentrations and upstream fluxes. And so, what can we do with this now, I guess, is sort of the question, or what does metabolism do with this? And to understand, I looked at just some simple modeling situations to really look at how fluxes could be controlled through small molecule regulation using these flux sensitive metabolites. So in an example here, we have two fluxes sort of entering a node and one leaving. So we have maybe, A plus B goes to C as a reaction here, or two different sources for the same product. And let's say we have one of these fluxes, or the product of one of these fluxes inhibiting the other. And so that sort of looks like this. We have, S arriving at a rate U. It's converted into this product, which is can also be generated through another pathway, and they both go. Then this product is used in some other downstream. If this reaction is reverse favored, what we can have is accumulation of this substrate, which then inhibits the other source. And so really what we can control by using this flux sensor as a small molecule regulator of some downstream enzyme or some parallel pathway enzyme is how much of that outflux V2 comes from one of these branches. And in the case where we have the unconstrained reaction, it's nearly impossible to control. As we change this upstream flux up to the maximum value, we can really only influence the partitioning, the source flux for V2 weakly. But if it is constrained, if this consuming reaction is reverse favored thermodynamically, then we can control, how much of this downstream flux V2 comes from U. As this flux changes. So the key thing is that it's as a function of the upstream flux. If you have a lot of flux coming in, maybe that's the prioritized pathway in terms of energetics or something like that for the cell. And so if you have a lot of flux coming in, a lot of the outflux will be from that pathway versus the other one. And so, it's just a small example of how you might be able to use a flux-sensitive metabolite to control flux distributions in the network, or rather how metabolism has evolved to do that. So, that's a local structure. I kind of called that one the feed-forward recombine motif because you have two fluxes recombining into one. But of course, there's a whole network of these interactions. And so this, it's called the small molecule regulatory network, or SMERN. I reconstructed the SMERN-free coli using data from Brenda, the allosteric database, et cetera, and other reconstructions, previous reconstructions, which exist in the literature. And this generated a really complex regulatory network with about 600 nodes, which represent reactions, and nearly 4,000 regulatory interactions connecting them. So, this is just a quick visualization of what that network looks like. You can see it's dense. There's a lot of different connections and subnetworks, very complex. And of course, because it's complex, it's sort of hard to characterize. So I looked to known motifs from systems biology and looked for those in the network and compared the frequency of those motifs to those from a randomly generated network that maintains the overall structure. And the typical motifs are, the classical ones are the feed forward loops. And the most basic of those have 3 nodes, and there's sort of 8 different ways you can structure those feed forward loops. And you can see when we compare those structures, the frequency of those structures in the real network to the randomized network, a couple of those structures jump out as being represented in extreme excess. And so the incoherent type 2, which is this one where all the interactions are negative, so it's inhibition interactions happening, and incoherent type 4, where you have some activation and some inhibition. These are really highly overrepresented in the network. So there's some clear structure happening here and some clear structure emerging in the small molecule regulatory network. So I'm not going to get into too much detail about why that is, but if there are questions, I do have slides after that we can look at. But I want to kind of keep talking about more how this structure is related to the thermodynamics of the network. So similarly to before, I characterized the thermodynamic constraint for reactions in these feed-forward loops to understand, is there some relationship between how favorable the downstream reactions are for the metabolites involved in these loops versus their role in regulation. And what you can see is that if we look at all the regulatory nodes in the network, the downstream the constraining thermodynamic parameter is significantly higher than if we just look at the network at large. So yeah, these regulatory reactions or reactions that produce metabolites that are themselves involved in regulation the consumption of those metabolites is thermodynamically unfavorable. And so, it sort of favors this idea that small molecule regulation has sort of arisen around metabolites which are sensitive or measure fluxes. It turns out, though, that the loop nodes are not special. They kind of, they have a similar distribution of constraining thermodynamic parameters compared to the rest of the sort of regulatory network. And in looking at where these loops exist in the network, I learned that, they're essential for stabilizing metabolic cycles. So again, I can get into a lot more depth with this, but I just wanted to give kind of a quick overview in the presentation. So if you have a cycle, where X is being converted to Y and Y is being converted back to X, which is very common in metabolism with some influx. There's sort of three different ways we can look at this. It can be sort of materially unconstrained. So we have a flow coming in, and that's going to drive the cycle. And some intermediates of that cycle might be regulating. That's what the dashed line represents here, that flow into the cycle. But we can also have an autocatalytic cycle, which is where X generates more of itself. So this is like the glyoxylate shunt in E. coli, for example, or a resource constrained cycle, where without some inflow, the cycle would collapse. And that's like the TCA cycle. And doing some local stability analysis, what we learned is that in the resource constrained case, you have to have positive coupling between the fluxes inside the cycle and those either entering or leaving. And then for autocatalytic cycles, you can have positive coupling as well. And the arrangement of small molecule regulatory interactions in sort of the TCA cycle glyoxylate shunt is flexible enough to allow both of those things to happen. And so this is kind of brings up an interesting paradigm that if we're going to be engineering cycles, we need to be thinking about the stability, and the stability not necessarily at the transcriptional control level, because cycles will collapse before gene expression can change. And so what this kind of uncovered is that there is a role for small molecule regulatory interactions in stabilizing those cycles by coupling fluxes, which means that you need a flux sensitive signal within that cycle. So malate and citrate, the metabolites I introduced before, are really great ones in the TCA cycle, for example, and they're involved in stabilizing the glyoxylate shunt this way, and also the TCA cycle. Great. So summary on that first part. The first question I asked was which metabolites make good small molecule regulatory signals. Those are substrates of what I call thermodynamically constrained reactions, or reactions which are favorable in the reverse direction. So substrates of reactions that would prefer thermodynamically to be running the opposite to the way that metabolism would like them to run. And how does small molecule regulation function to control fluxes? Well, feed-forward regulatory structures can distribute and stabilize those fluxes. So we saw that with the motif, where we could control how much of some outflux came from some source versus another. And then also in stabilizing cycles with feed-forward regulatory structures. But in addressing these questions, a new question naturally arises: what makes small molecule regulation a good mechanism for interpreting flux sensor signals? So why this mechanism and not some other mechanism? So to answer this, I consider the trade-off inherent to flux sensitivity. Metabolites which are most sensitive to upstream fluxes have concentrations which are the least sensible by downstream enzymes, because flux sensitivity arises from constrained operation near saturation. So as I said before, if we have this very simple case where a substrate is arriving at a given rate, if it's operating well below, the system is operating well below saturation of the consuming enzymes, then as more S accumulates, that's fine, it can be consumed. And so the steady state concentration may not be affected by the changes to this upstream flux. But in the case where S has accumulated, now we have a constant rate at which it's consumed. So changes to this upstream flux are going to cause changes to the substrate concentration, which will be decoupled from downstream changes. Okay, so if U goes up, V has nowhere to go because it's already at Vm. And so even though S will go up as U goes up, that change might not be sensible locally in the network. So this is where we may have some inhibition interactions occurring, or small molecule regulatory interactions occurring. And so to characterize this, I considered the sensitivity of catalysis to changes in substrate concentration using what's called elasticity or scaled sensitivity. So an elasticity is just how much does this parameter V change with respect to a change in S scaled to their steady state values. And for simple Michaelis-Menten kinetics, we can get a closed form, solution to this, and it just looks like the Michaelis-Menten parameter divided by Ks plus S here, which means that as this substrate goes to saturation, the sensitivity drops to 0. You can see that in this curve here, and that's for catalysis, simple Michaelis-Menten kinetics. We understand this because we get to Vmax, right? And at that point, changes to the substrate concentration are not going to change the rate at which the enzyme can operate. In contrast, for inhibition, the sensitivity is maximized when the inhibitor accumulates. And so in this case, we're looking at inhibition, simple, I think uncompetitive inhibition is what I used here, but you can really use any mode and you'll get the same kind of qualitative results. And here eta is the Hill parameter. So you can see that the sensitivity goes towards eta as saturation by the inhibitor, as the inhibitor reaches a saturating concentration. So the important thing here is that these curves do the opposite thing. So as we saturate an enzyme by its substrate, that same substrate, if it's an inhibitor, will be, or what it inhibits will be maximally sensitive to changes in that substrate concentration. So when you consider this in the context of an open system with, again, a thermodynamic constraint, that very simple case, S to P and P being consumed, it's clear that the constrained consumption favors the role of the substrate S as an inhibitor, because above a threshold, and this threshold is just chosen sort of arbitrarily here just to show the behavior, but you can see that above some value, the sensitivity to changes in the substrate drops to 0. Or close, or is minimized anyway, whereas the sensitivity via inhibition is maximized. So these the mechanisms do opposite things essentially, and we can kind of consider this as a measurement problem. There's 2 ways of measuring S, each with unique constraints, and we can find those constraints which describe how information can be transmitted by each mode. So if we have a signal, which might be a concentration, it carries a certain amount of information given by H here. We put that through a constrained measurement process, like catalysis by an enzyme with Michaelis-Menten kinetics or some kind of saturation kinetics versus regulation. And here inhibition, we can see that we either maintain that signal or we reduce the information that's contained in it. And these do, again, these mechanisms, catalysis versus inhibition, they work in opposing ways. And so we can actually calculate a constraint on this. My apologies, this should be greater than or equal to, not equal to, but it's very sort of simple to calculate the information loss in terms of the upstream. The information about the upstream flux is given by this simple logarithm where A is the concentration of a substrate or inhibitor. And this is what this curve, this constraint curve looks like. As A goes to 1, we lose all the information, or we lose none of the information rather, but as A goes to 0, we lose all of the information. And the key thing here is that catalysis moves us this way along the curve as the substrate concentration increases, whereas inhibition moves us this way along the curve as the inhibitor concentration increases. So enzymes nearing saturation will lose all of the information about the upstream flux because they're at saturation. But if that substrate is used as an inhibitor, we can have 0 information loss in theory. Of course, this model doesn't include any noise, and I can talk about noise a little bit in the questions if that's sort of interested. Yeah, if people are sort of interested in that. Now, on the flip side, enzyme activation functions similarly to catalysis in that the sensitivity drops as the activator concentration reaches saturation, and that's same as a function of the thermodynamic constraint. So what this means is that activation maximizes information transfer in the same conditions as catalysis. And so this really would suggest to us that inhibitors should be associated with strong thermodynamic constraints. And they should also be consumed, they should also be associated with many consuming reactions. Yeah, and indeed we find that. So if you look at the distribution of inhibitors, the thermodynamic constraint is significantly higher than that for sort of non-regulatory reactions in the network, and the degree is also much higher. And then what we also find is that the previous thermodynamic threshold that I suggested of -4 is actually a really good indicator of sort of inhibitors in the network, where we can see that inhibitors are enriched among reactions which fall above that thermodynamic constraint compared to the network at large. And then finally, sole activators are rare in the regulatory network, and that's because they don't solve the same problem that inhibitors do. And in fact, of the inhibitors in the network, which I reconstructed, almost half were also activators. And so in terms of the information preservation, these metabolites are still doing the same, are able to achieve that while also acting as activators. And there's really only 20 reactions that are sole activators in the network. Okay, so I mean, this is sort of, I know, pretty, in some ways pretty complex. And so I want to try to kind of bring it down to a concrete message and how that relates again back to sort of developing new synthetic biology tools. The key messages here, substrates of thermodynamically constrained reactions, they carry information about upstream fluxes, and that information is measured in different ways by inhibition and catalysis in the network. And the organization, the SMERN itself, the network is organized around maximizing the amount of information about fluxes that can be pulled out of these concentrations. And so, I mean, I just sort of have a representation of that here. Metabolism under different conditions you might have, which are, those conditions are represented by different flux distributions. You might have different concentrations which respond to those different fluxes, right? So the yellow metabolite is, you can see, steadily increasing as these fluxes change. And that can give us some relationships between concentrations and fluxes. And really what the small molecule regulatory network does is sort of disentangles those relationships to separate into different discrete states, or maybe not quite discrete, but different states where those metabolites are able to regulate different flux distributions. Okay, so bringing this to synthetic biology, what does it mean? I actually started my PhD work on a pure SynBio project, and I ended up here. And so I'm always thinking of how can we use this kind of thing to really inform what we do as a synthetic biologist? Well, a major goal of SynBio projects is to rationally redirect metabolic flux towards desired products. We generally create transcriptional regulatory circuits to achieve this. And we might be able to use these sort of flux sensing metabolites to do that. So a good example is sort of, if you have a product X that has two different precursors, and you need to balance the rate at which those precursors is produced, so that, for example, an intermediate doesn't accumulate, maybe it's a toxic intermediate, then how can you create a regulatory structure that will allow you to balance those two rates? Well, what I've done is show you that maybe D, this intermediate, is the optimal signal for that regulatory relationship because it's a substrate of a thermodynamically constrained reaction. So really what this work is about is finding what signals are good so that we can build circuits around using those signals. And this is be what it looks like. And we can also use what's already been developed. So for example, there's already a fructose 1,6-bisphosphate biosensor, which reflects glycolytic flux and has been used to give a fluorescent output as a function of the upstream flux rather than a concentration. And there's at least one RNA-based FBP biosensor, and RNA-based sensors, of course, are adaptable to many different metabolites. So you might have a heterologous pathway with a very interesting or useful metabolite that a biosensor could be developed for to balance those fluxes. And what that might look like, just as a sort of very simple representation, in your pathway, if you have low or no flux, you have no or low expression of a particular enzyme or a circuit. Whereas if you have high flux, you can then have high expression of that circuit. And obviously, this is a very basic kind of imagined structure. There's so many different ways that you could use flux sensors to create or to as input signals to gene circuits. And so I hope that I've kind of opened up the idea to finding those signals and finding the optimal ones for these kinds of circuits. And so with that, I would like to open up to sort of any questions there might be, further chat, etc. Thank you.
Wonderful talk. Great. So,
Thank you.
I love it because not many people see metabolism in that, angle, and then you brought up very unique kind of perspective to the metabolic engineers. I believe many metabolic engineers will benefit from that one.
Hopefully.
So I have questions. So I see, utility of those approach, but still, I'm thinking about something bigger in terms of kind of consortium engineering. And then I also heard, species and species also interact somehow by exchanging metabolite. And then to basically model, even two species interaction, I heard that is very, very challenging. And I want to hear from you, your perspective, what is really the challenging part regarding, or to make sure, those interactions kind of well studied, at the same time, eventually making huge consortium modeling network, called metabolism network. So can you give some insight?
Yeah, I think maybe my understanding is that scaling metabolic models is computationally challenging, and this is part of the reason why FBA as a sort of tool set is so attractive. But ultimately, FBA and even DFBA is built on a really flawed assumption of steady state. And even for a single organism grown in a batch culture, that steady state exists, that is described by FBA, exists for a fraction of the time of that culture. If you have a chemostat, of course, it's a better representation. And I think the challenge is that when you have a consortium of species, you're just fundamentally, you're never going to have the steady state really that you're looking for that is easily modeled by FBA. And so then you have to look at kinetic modeling. Kinetic modeling has so many details. There's a lot of hidden variables that you can never know. And computationally is very expensive. And so I think that is sort of the major challenge, is that communities, there's a level of dynamism in bacterial communities or microbial communities that is extremely hard to capture. And in terms of our observations, translating our observations to that, and extremely hard to model. And so I really do think that this is where, as metabolic engineers, machine learning and AI approaches will be really excellent for us. I think that that's like where we should be focusing those efforts. I also wonder about the utility of actually building consortia and how difficult that is. Is it worth it? I wonder if there are specific products where that might be useful. And one thing that we talked about in Krishna's group often was sort of a thermodynamic niche, that some organisms are there to kind of get the top end of the Gibbs free energy of the metabolites, and then some organisms are there to get sort of what's left after they're done. And I think that kind of modeling also is a way to maybe simplify what's going on in consortia. And but again, the challenge is thermodynamics are not really well characterized for metabolism, especially for cycles. I think cycles are really difficult. It poses a real challenge to modeling thermodynamics in metabolism and therefore in consortia.
I see that makes sense. Then thank you so much. So in fact, we already over my time, 11:05 p.m. I actually 12 midnight, midnight. So I want to close.
Okay.
But I love it. I mean, so we probably chat more after this official session. So thank all for joining and staying today. And due to AICT, let me see. No. I'm sorry. I'm still so sleepy. I'm looking at something else. So we'll meet again next week on November 3rd, Thursday, the same time, the same Zoom link. We'll have Dr. Kelly Rose from American Society of Hematology and Professor Jennifer Brophy from Stanford University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. And thank you. I'm stop recording. Okay, give me 1 second.